So this is me. I'm just started my third year of my PhD, which means I'm getting to the point now where I really need to finish up all my experiments and make sure that I've got all the answers to everything and I've cured everything so that I can start writing up my thesis. So this is the Malaghan Institute here. Some of you may have heard of it or even been to visit our building before. Based over the harbour here, it's got this beautiful view out the window, which as a PhD student, the best you can hope for is to have a nice view out the window because you don't make it out before the daylight is gone. So the title of my talk today, oh, that's my little cry. The title of my talk today is developing a therapeutic vaccine for allergic asthma. So I'm just going to talk to you about what I've done so far. So my talk outline, I'm going to give you an introduction to asthma and allergic airway disease, but as Grant suggested earlier, probably a lot of you have already know much about this disease and you may know people who are affected or it may be your own battle. I'll then go on to talk about some work that I've done so far in terms of developing a vaccine in the lab and the things that I'm currently looking into. So just as a broad introduction to asthma, New Zealand has one of the highest prevalences in the world for being affected by asthma. Nobody's incredibly sure why, because we have quite good health systems here, we have very clear air, none of the usual factors that you would expect to be causing high levels of asthma, such as pollution or poor living conditions, although a number of New Zealanders do live in quite damp conditions depending on where their housing is. But we have 15 to 20% of us who are affected by asthma. So that's a really huge statistic, and there's probably a number of people in this room who will know people who are affected. There's many different types of asthma. So asthma can be induced by things like exercise. Some people get occupational asthma. But the asthma that I'm going to talk to you about today, the one that I focus on, is asthma that's developed due to an allergic reaction. So this is just a picture of what asthma actually does in your lungs. So we have the lungs here and we've zoomed up on some of these airways. So this is a normal airway here. You probably can't see the words on the screen there very well, but in the normal airway you've got clear air space so that the air can get through so you can oxygenate your blood in your body. Basically in asthma, a number of factors come into play to cause this reaction, including the smooth muscle around your airway contracts, tightening the airway. You get an increase in mucus production inside the airway lining, and you also get the infiltration of a lot of immune cells which cause inflammation. So all of those factors come together to cause this restriction of the airways, making it hard to breathe. So in terms of the actual allergic process that develops into asthma, we have a process going on in our body all the time of immune surveillance. So one of the key immune cells is this cell here called the dendritic cell. Now the job of the dendritic cell is to circulate in the blood and also lives in different tissue compartments and organs in the body. And its role is to sample everything it comes across and decide whether or not what it has come across is a threat to the system. So in the case of things like bacteria, viruses, infecting organisms, you want this cell to recognise that that's a threat to the system and to instruct the rest of the immune system to carry out a response. However, in the case of harmless things, dust and pollens, you want the immune cell to recognise that this is not a huge threat to the system and not to carry out an immune response. So the normal scenario for harmless things that we inhale is that these things come into the lungs, they're recognised by this dendritic cell. The dendritic cell then presents it to other members of the immune system, primarily this cell here called a CD4 T cell. And during this interaction, the dendritic cell would have the appropriate response of telling the cell, if you see this, it's harmless, don't have a reaction against it. That would lead to no allergic response in the lungs and tolerance within your immune system. However, in the case of allergic asthma, the same process occurs, But when the dendritic cell presents it to the other immune cells, it presents it with other associated signals which tell the immune system that this is a threat and you need to carry out a response. So the immune system launches this response to that specific allergen, and you get allergic airway inflammation or allergic asthma. So this is just focusing down even lower in the system to the specific disease-mediating cell, which is our CD4 T cell here. Now the mechanism of action of this cell is it produces a number of chemical mediators, and it puts these chemicals out into the system, and they have effects on various parts of the lungs and the airways, which result in the characteristic features of asthma. So one of these chemicals that it produces is called IL-5 or interleukin 5, and that chemical acts on the immune cells called eosinophils. So eosinophils are key cells in allergic reactions. They produce a whole lot of other immune mediators which cause widespread effects. The effects of eosinophils and other immune cells lead to mucus secretion in the airways here and airway hyperresponsiveness, which basically means that muscle contraction around the airways. So my work is focusing on developing a vaccine for allergic asthma. The current treatment we have is corticosteroids, which many of you will have heard of or potentially experienced. And the mechanism of action of corticosteroids is that they go into the lungs and they effectively stop the immune response of all the cells that are causing inflammation. So it effectively prevents that exacerbation of disease. But they're non-specific, so what that means is that they also affect other immune responses. So it means that you may have a number of side effects, and also may mean that when you come into contact with, for example, flu infection, you might be more susceptible to disease because you've got this drug which is dampening down your immune response. So what my work is focusing on is finding a way that we can develop a more targeted therapy that only knocks down the immune responses that we don't want to be occurring without touching the important immune responses. So there were two questions that we're primarily asking in our development of this vaccine. Could we develop a vaccine which could prevent airway inflammation from occurring? And could we have a therapeutic vaccine which is effective when people have already been suffering for asthma for some time? So with our first question, can we use a vaccine that can prevent airway inflammation? The normal scenario that you would think of when you're thinking of getting a vaccination is that you get treated before you get a disease. So, for example, for measles, mumps and rubella, we have a vaccination in New Zealand which gets given when you're a child, and that means that when you come into contact with either measles or mumps or rubella, when the bug gets into your system's got that prior knowledge and doesn't have has an immune response stored up ready to go, so that you don't get affected by that disease. So the first question I was asking is, can I give a vaccine before someone develops allergic asthma to prevent them from getting it? So I have some results here from some lab experimental models of allergic asthma, and it looked quite positive in terms of pre-treatment with a vaccine. So in this graph here, I'm just showing the total cell number that has infiltrated into the lungs. So you'll recall from the first slide that one of the key promoters of inflammation in the lungs is all of these immune cells flooding in and producing chemical mediators. So we have our asthmatic lung here, this blue bar, and so you can see there's a large number of cells infiltrating into the lungs in the asthmatic environment. This here is a healthy lung, so there's hardly any cells in the normal state. In the experimental model that was treated with the vaccine, you can see we've got a huge drop in the total cell number in the lungs. And the key player in the allergic response that I mentioned earlier, the eosinophils, we also have a huge drop in the eosinophils. They're almost absent. So this means that there's a really low level of inflammation going on when the contact with the allergen occurred. The chemical mediator that I mentioned, IL-5, we can also see a slight decrease in this mediator, which means that it's being stopped at the early stage of the response. In terms of some histology here, these are actual pictures of lung tissue during the disease state and the healthy state. So in a healthy lung here, you probably can't see a lot of fine detail from the back, but what we're basically looking at is the air spaces. So you can see there's a lot of clear space here. Here's a larger airway and these are smaller airways. So there's a lot of clear space in between the airways, so there's not this huge infiltration of immune cells. In the asthmatic lung, on the other hand, you can see all these tiny little dots. That's a whole lot of immune cells that have just infiltrated and causing a whole lot of congestion in the lung. When we give a vaccine before the allergen exposure has occurred, you can see that the airspace is also quite nicely clear. Now the frames down the bottom here are stained with a slightly different chemical which shows up dark purple granules where mucus production is occurring. So in our healthy airway, you can't see the staining for it. In our asthmatic lung, you can see this airway here is hugely affected and has massive mucus production. And in the airway that has been treated with the vaccine, the mucus production is significantly decreased. The next question we wanted to ask is, could we develop a therapeutic vaccine, or could the same vaccine be used in a therapeutic context? So it's easy enough to, well, it's not necessarily easy, but it has been known to work really well to prevent illnesses before they occur. But that process that I showed earlier of the allergic reaction being carried out is a really strong process. So once it's been occurring over a long period of time, you get changes in the airways and in the lungs that take a long time to resolve. So the question is, could we come in with our therapy and promote resolution of the disease after it's been occurring? So looking at similar parameters here, we've got the cell numbers that are infiltrating in the lungs. So between our asthmatic lung and our vaccine-treated lung, there's a huge drop in the total cell numbers, and in the eosinophils, again, there's a huge decrease. IL-5 is again slightly decreased there, showing that it's in the early stage of the response. And similar histology pictures here. You can see that when treated after asthma has been occurring, the healthy lung is clear. We have congestion again in our asthmatic control. And in the vaccine treated, we've got a lot of clear space appearing again. Similar results with our mucus production. The asthmatic one here is highly affected by mucus, whereas in the vaccine treated, we have a big decrease. So we're pretty excited about those results. I think that this is quite a positive point to start from with our therapy. It looks like it's really promising in terms of both preventative and therapeutic effects. And the first 2 years of my work has just been looking into characterising that, optimising it as far as the lab conditions are concerned, and looking at ways that we can make it the most effective. What we don't know yet is how exactly the mechanism of action is occurring. And before you can go into the clinic and start giving this to patients, you need to understand what processes your vaccine is working by, what things it's affecting in the immune system, what other potential side effects may occur, so that we're not just going in there and throwing something in and thinking, oh, it looks good in this context, but actually what does it do in a number of other contexts. So future work that I'm working on is looking at how it works, but we do have a theory at the moment of how it's working. So if we look at our process here of airway inflammation, we've got the dendritic cell presenting this allergen as a threat to the immune system, and that's causing airway inflammation. We know that our vaccine stimulates another immune cell called the cytotoxic T cell, or CTL. And what this cell does is that it can also recognise allergens being presented on the dendritic cells. What the response of that cell can then be is to kill the dendritic cell. And when it kills the dendritic cell, that prevents the presentation of the allergen to other cells of the immune system and stops airway inflammation. So it's known that this process occurs on a very small scale in a number of individuals in the normal immune system, and it's thought that this process is occurring to limit immune responses so that the immune system doesn't overreact to any sort of threat. So it's a normal physiological mechanism that is there to basically protect you from your immune system going wild. Many of you might have heard of autoimmune diseases, where your immune system overreacts and causes all kinds of different diseases in the individual. So we know that while the immune system can be incredibly positive and really useful for us at warding off disease, it can also have really negative effects on our system if it's not kept in tight control. So this phenomenon is thought to be a normal mechanism, but as I said, it's only there in a really small amount. So what we were thinking with our vaccine is to increase this population and see whether or not in increasing that population we were able to cut out this allergic response. So what we're working on at the moment is that question, how is the vaccine working? We know that it's stimulating those cells, and it seems that it may be that that process is occurring, that those cells are killing off the dendritic cells and preventing the initiation of the response. We also have done some work in testing this vaccine alongside corticosteroid treatments, and we know that it is less immune suppressive than corticosteroids, so that's positive in terms of it being a targeted therapy. And we have a number of questions about what may happen in the clinical context. For example, are these cells active in patients who don't have allergy? So the huge question in the research world of allergy is, why do some people get allergies to things that other people don't get allergies to? And there's a number of theories, but nothing really conclusive so far. So one of the questions that this experiment has raised is that potentially these cells are present and active in healthy people, and they're controlling this response and preventing allergies from occurring. But potentially in people who have allergic asthma, this population may not be there. So clinical tests have not been done yet to test various people to see whether or not they have these cells or don't have these cell populations. But if they were not present in allergic sufferers and we could promote them with our vaccine, that could potentially be a way that we can carry this into a clinical context. So I'd like to say a huge thank you to the Rotary Club of Wellington for supporting my PhD research so far and continuing to support me in my final year. It's really great. I was saying earlier to Tony that I've only ever heard really good things about the Rotary Club, and that they're so supportive of a whole range of things that are going on in the community. So yeah, I'd really like to say my sincere thanks that you've chosen to support me. I also need to thank Health Research Council, who gives additional funding for our research, and the Malaghan Institute of Medical Research, where I carry out my research under the supervision of Professor Franca Roncaiza. And these are the names of my group members who help me with big experiment days, so that sometimes I do actually get to see the daylight. Yeah, we have some information sheets out the front on the table where you would have got your name tags. So if you're interested in finding out anything more about the Malaghan Institute and the research that we do there, feel free to take any of those pieces of paper. There's also a sign-up sheet there where you can put your name down if you'd like to receive our newsletters from us every month. Thank you. Most people who are allergic to one thing, as you say, are allergic to a number of things. So our targeted therapy is looking at one specific allergen to begin with. For example, a house dust mite allergen is one of the key ones we're looking into. So that will be targeted against that response. But what we are finding at the moment, and I'm actually carrying out an experiment which is finishing in the next 2 weeks, is that promoting those CTL or cytotoxic T cells also has some effects on dampening immune responses to other allergens. So it could be that it is reducing the population of dendritic cells that are driving a number of allergic processes. But as I said, the experiment results in the next 2 weeks might give me some more information on how effective it is for other allergen specificities. So I've presented my work at conferences in Australia in the last 2 years. I'm yet to do my international tour. That'll be in March next year when I go and look for postdoc opportunities. But yeah, generally we've had a lot of good interest from international collaborators that I've talked to at the conferences in Australia. And it's actually quite different what we're working on here. A lot of people are working on using dendritic cells as their vaccine, and they're trying to prevent these CTLs from killing them because they want the response that the dendritic cell is carrying out. And we're using the complete opposite. We want to promote the response of these dendritic cells being killed. So it's quite a novel idea that has come out of our lab. So it's interesting to a lot of people and also kind of shocking because they're used to trying to prevent that response rather than to harbour it. Yeah, so essentially the problem I was talking about with the current treatment, corticosteroids, is just that. It does wipe out a whole bunch of immune responses, both deranged and useful. We're hoping that with this targeted therapy, it's only going to kill off specific cells. So there's an immune response that is carried out there. With my little cartoon pictures, it didn't look all of that specific. But basically, the cytotoxic T cell can recognise dendritic cells that are only presenting one specific allergen, and you can make them of different allergen specificities. So the idea is it will only kill a small subpopulation of the dendritic cells, and just the ones that are specifically presenting that allergen as a threat. So those treatment cells won't be able to recognise any other dendritic cells that are presenting, say, different viruses or bacteria. That's the idea. I'm yet to test whether or not any other immune responses drop off, but that's definitely something that we need to focus on before we take it into the clinic. I'm not sure if anyone here has heard of Malaghan innovation. Some of you can. So we work alongside Malaghan Innovation. They have developed a number of chemical compounds and a number of drugs for projects that we're working on at the Malaghan Institute. So there is some talk going on at the moment with Malaghan Institute and myself and my supervisor with our work. And they are working on something that can promote this response, and I've just finished preliminary testing of that drug. So that would definitely occur in clinical trials. The problem is that the experiments that you do on a lab bench can be quite different from what happens in the human population. Because humans are so widely varied in their responses and whether their immune system is particularly strong or not as strong as others. So it would be difficult to predict how things may go in the clinic. And in terms of dosage, that would definitely be something that would be worked out at that time. But yeah, you really don't know until you get into clinical trials. There's been lots of preliminary research that looks really promising when it's in the lab stages, but so many of those drugs get into the clinic and they fail because they haven't been able to overcome the systems that the human body has in effect. So hopefully this one will be effective, but yeah, that'll be worked out.